Yesterday you heard from Donal about piRNAs, the short RNAs that are involved in protecting the germline cells from transposable elements. There are several other short non-coding RNAs that I'm just going to mention here. MicroRNAs are ones I'll talk about today, which usually modulate gene expression normally and during development and in disease. And then tomorrow I'm going to say a few words about siRNAs and algoproteins, how they interact with these things. And siRNAs are very, you've already heard about them. They're already, they're used in inactivating messenger RNAs during development and also in experiments, very useful tools. So today I'll focus only on the microRNAs, and I know that there are some people in this audience who could probably give this talk better than I can, so I apologize to those of you who, well, at least as well as I can anyway. I'm going to give, first of all, an overview of the microRNA synthesis and functions. Then I'll use an example of microRNAs that function early in Xenopus development, and then as diagnostic tools in disease. Just a bit of review here. MicroRNAs are short molecules, about 22 nucleotides long, encoded in the genome. Human cells have anywhere from 500 to 1,000 different microRNAs. A lot of viruses bring microRNAs in or bring in the capacity to synthesize their own. They associate with proteins, Argonaute proteins, to form complexes. It's really the protein complex that's important, and these are called RISC, RNA-induced silencing complexes, which silence the expression of messenger RNAs. And the targeting of messenger RNA is through base pairing with regions of the microRNA with sequences that are in the 3' UTR of the message. And that recruits a bunch of proteins. The biogenesis of microRNAs, they can either be made as single molecules or precursors with multiple molecules encoded in the same precursor. These are in hairpin regions that get processed within the nucleus by an enzyme called DRASHA with the cofactor DGCR8 down to precursors, pre-microRNAs. You can see these are imperfect hairpins, which are exported into the cytoplasm using exportin 5 in RAN-GTP-dependent manner. And in the cytoplasm, another enzyme, dicer, along with some cofactors, cleave these pre-microRNAs into duplexes, symmetric duplexes, one strand of which is selected to be incorporated into the RISC. And then this is the sort of the business compound that's going to actually bind to its target. Each one of these steps is controlled, and you can get control at various levels depending on the microRNA. MicroRNAs often affect deadenylation as well as translational inhibition. In the deadenylation experiment, one sees that the microRNA will bind to its target in the 3' UTR, recruiting several other proteins. GW182 is a sort of a central player in this, and that in turn recruits deadenylation machinery, and you can see the poly A gets shorter, and ultimately the RNA is degraded. There's also, not shown here, that there is also translation inhibition that you've already heard about as well. Of course, translation of this molecule is pretty inhibited too, so this no longer exists. So we've been studying microRNAs in our friend Mrs. Xenopus here. Marv has already introduced you to the frog oocyte. The oocyte matures into an egg by progesterone on fertilization. the egg starts to divide very rapidly, and as Marv told you this morning, there's essentially no transcription that occurs until around the mid-blastular transition, which is about 7 or 8 hours after fertilization. By that time, the cell division has gone through about 12 cell divisions by that time. So there's about 4,000 cells in the embryo. The embryo has been working very fast, very hard, making DNA and protein, but no RNA, or very little RNA. But we noticed early on that slightly before the mid-blastular transition, where there's this big change in transcription and messenger RNA stability, a molecule did start to accumulate, and that's a precursor that we identified as pre-microRNA 427. 427 precursor is matured into the mature microRNA, and this accumulates in very high amounts very quickly in a very short time. And so we asked what might this molecule be doing, and you can see one candidate, and one that we think is the real target here, is that several maternal messenger RNAs undergo a transition at this point. Marv, again, emphasized the importance of the maternal messenger RNA in the early embryo. And here we see that here's this is actually a chimeric RNA that contains the 3' UTR of a cyclin messenger RNA. This molecule gets, this reporter RNA gets polyadenylated with time, and then it undergoes deadenylation and is destroyed. So we thought that perhaps this microRNA might function in that deadenylation. And so we tested the role of 427 by injecting either pre-mir427 into the embryos, or we could put in target messenger RNAs into the embryos. And that's the gist of the kind of experiment we did in this series here. I mentioned that the cyclin B2 messenger RNA undergoes deadenylation, and the experiment to show that is shown here. And I think we're going to run into some troubles because of the shifting in between computers, but I think this will work anyway. This is the region of interaction between the microRNA 427, which should be strung out this way, and a sequence that's in the 3' UTR of this messenger. These are elements that Marv already talked about this morning, CPE and earlier today, and the poly A, A2UA3 sequence. Now, microRNAs interact with their target through a region called the seed sequence, which is sequences near the 5' end from position 2 to about 7 or 8 in the RNA. And that's a perfect base pairing. And then there are imperfect pairs between the microRNA and its target. But it's the seed sequence that's the important region. So in order to see whether this interaction was important, well, first of all, here's just what you saw before, but quantified to show that the RNA really does disappear. To see whether that sequence is important, we mutated part of the seed sequence there, and now you see that the RNA, the target, is stable, doesn't turn over. So that indicates that this sequence in the seed target plays a role. To show that this really is a direct interaction with the microRNA that's responsible, this is just what you just saw, that the resistant mutant here. We've injected into the embryo a pre-mir427, where we mutated sequences in the microRNA so they would be complementary to the mutated form of the target. And now you see that the RNA that was mutant RNA that was stable is now subject to deadenylation. And this deadenylation in the presence of the exogenous microRNA precursor is specific because if we just put in the precursor, the wild type precursor does not accelerate deadenylation. It's only the compensatory mutant that does that. The question now was whether the microRNA sequence itself is important, or is it the proteins that it brings in that are important? So what we've done, this is just a repeat of what you've seen. Here's the wild type gets deadenylated. This is the mutant form of a target is stable. And then we put in the compensatory mutant. You get deadenylation. Now we've repeated this experiment, but in the messenger RNA of the target, instead of having a sequence that's complementary or partially complementary to miR427, we put a sequence in that would be complementary to another microRNA, LET7. And here is the sequence that we put in. Now, it turns out that these embryos do not have any LET7 microRNA in them. So when we put this target, this reporter RNA into the embryo, it's stable. It's not degraded, deadenylated. However, if we put into the same embryos, also inject the precursor to LET7, pre-LET7, now you can see there is deadenylation. And that deadenylation depends on the sequence of the precursor because the wild type doesn't, the 427 sequence doesn't compensate. So we conclude that the deadenylation requires the protein, but not the RNA interaction. Now, we've taken, or I mentioned before, the importance of seed sequence. That's really the region that interacts with the main region that interacts with the targets. We've looked in a variety of messenger RNAs to see if there are comparable seed sequences in other organisms. And we can see them in frogs, fish, humans, etc. Or look for the presence of targets for this kind of seed sequence. And in all cases, we find that, that the microRNAs that we see in frogs that I've just been describing here, the same seed sequence can be found in fish and all these other organisms. The others, there's also in mice too. And in all cases, the tissue that contains these microRNAs are embryonic tissues, early embryo placenta, or embryonic stem cells. So we're tempted to think that this might be sort of an ancient system that was that evolved to remove or to downregulate maternal messengers or messengers that have to be removed early in development. Just a little history here. Two other, or LET7 was really and LYN4 were the beginning of the sort of the study of microRNAs, and it was shown that this was absolutely essential for early development in worms. There are a number of labs now, Bozzoni among them, looking at microRNAs in heart and other tissues, normal development. And much as these microRNAs are important in development, they don't seem to be important for survival of embryonic stem cells. So it's more of a switch, at least in this case here. If they're so important in development, we asked whether they might play a role in cancer, where the development gets out of control and the cells start to divide too rapidly. And in this case, we collaborated with Tang at Cornell, and we found that one particular microRNA, 155, now this is in human lymphoma cells. These are primary lymphomas that were isolated and biopsied. The miR155 is elevated in human lymphomas, and these are graded according to the severity of the lymphoma. And as you can see, it's the red bars that are important here. These are the levels of the mature microRNA 155 that we assayed, we could pick up in different lymphoma biopsies. So the more aggressive lymphomas, like the activated B-cell lymphomas, are the ones that really have the elevated 155. You can see that the blue indicates the precursor of the 155. That also goes up, but not nearly as much as the mature RNA itself. So the take-home message from this is that the levels of 155 are several-fold higher in the more aggressive types of lymphomas. And so this is a possible indicator of some diagnostic tools that we and others are developing. The targets for 155, this is an earlier slide that certain transcription factors are good targets for the interaction with the seed sequence. Now, a number of these have not been verified experimentally, but some of them have. And a group that's in David Baltimore's lab have shown that a number of transcription factors and factors that control cell division are definitely regulated by 155. 155, miR155, seems to be involved in the initial response of the innate immune response, among other things. So it's an early player in the immune system. This is just a very quick slide. Actually, it's a fairly old slide, but it's just to show that a number of different tumors have been correlated with elevated or reduced amounts of different microRNAs. And this is a really small sampling of what's now known. In some cases, the microRNA goes up. In other cases, it goes down. The phenotype also changes with whether it goes up or down. LET7 and 155 correlate with the survival in adenocarcinoma. LET7 is known to interact by work that Frank Slack did. It interacts with the RAS gene messenger RNA. And by having low LET7, you get a high amount of RAS, which is a proto-oncogene, and that it makes sense that you can get a tumor of that sort. So now this is just with, in cancer, it's, as I've mentioned before, there's a lot of other biology going on now with these as indicators of what's going on in healing, wound healing, and so forth. So just to summarize, that microRNAs often control gene expression in normal cells during development and disease. And the one remaining question that I have is why did it take it so long to find this out? Because they're so central to our understanding. So tomorrow I'm going to talk about not this sort of thing, but the siRNAs and complexes formed between siRNAs and the oligoproteins. So with that, I'll stop. Thanks.